In this video, we're going to be looking at the light-dependent reaction during photosynthesis. There are three key points to bear in mind when we're looking at this reaction in general. The first is that light is absorbed by chlorophyll that is found in the thylakoid membrane. Energy from the sunlight absorbed is used to form ATP, and hydrogen ions from water are used to reduce the coenzyme NADP. Some structures found that we need to think about are the stroma. The stroma is a colourless fluid surrounding the grana within the chloroplast. Within these grana are stacks of thylakoids. The membrane that we're going to be looking at is the thylakoid membrane. Inside of the thylakoid membrane is the lumen. Light-dependent photosynthesis in general can be split up into two sections: non-cyclic photophosphorylation and cyclic photophosphorylation. The first stage in light-dependent photosynthesis that we need to think about is how two electrons are generated that enter photosystem 2. This is done using a process called photolysis. Photolysis is where one water molecule is split into two hydrogen ions and an oxygen ion, releasing two electrons. This process provides low energy electrons which are used in photosystem 2. The oxygen produced in this reaction is released by plants during photosynthesis. Light energy is used in photosystem 2 to raise the electrons from a low energy state to a high energy state. This electron is then accepted into the electron transport chain. An electron transporter called plastokinine accepts the electron from photosystem 2. The shape of the plastokinine changes when it gains a charge from the electron, which allows it to move across the thylakoid membrane. The electron carrier donates its electron to the hydrogen pump. The energy is used by the proton pump to pump protons into the lumen from the stroma. The energy from the hydrogen electron is used to pump hydrogen ions through the hydrogen pump. In part, this is due to the electrical charge as we move down the hydrogen ion channel, changing as the electron passes through it. As the electron passes through it, that gives the channel in the middle of the hydrogen pump a slightly negative charge, which in turn will draw the positive hydrogen ions through. Once the electron has moved from a high energy level to a low energy level in the hydrogen pump, it is accepted by an electron acceptor which carries the electron to photosystem 1. The electron is then accepted by photosystem 1. Light energy is absorbed by the chlorophyll. The electrons are then excited so therefore raised to a higher energy level. The electron that moves through photosystem 1 is accepted by the coenzyme NADP+, which is reduced to NADP. Hydrogen ions move down the concentration gradient through an enzyme called ATP synthase, which in turn will produce ATP. The ATP synthase catalyzes the production of ATP by adding an inorganic phosphate molecule to ADP. In cyclic photophosphorylation, an electron is accepted by photosystem 1. Light energy is absorbed by the chlorophyll, so the electrons in chlorophyll are excited, so therefore raised to a higher energy level. The electron carrier donates its electron to the protein pump in the membrane. This protein pump is used to move hydrogen ions across the membrane by active transport. The hydrogen ions move down the concentration gradient through the enzyme ATP synthase and provide the energy for ATP synthesis. As before, ATP synthesis involves adding an inorganic phosphate to ADP. The electron is recycled from the hydrogen pump back into photosystem 1 and is not used to reduce NADP+. This is why the second part of photophosphorylation is referred to as cyclic. I hope that this video has been useful. Please feel free to go back and watch it again and pause it at any stage. In the links below there is a downloadable diagram that you can fill out whilst watching this video a second time. Good luck in your exams.